Hello. Last lecture we talked about gastrulation and the stage which follow blastulation, and the main aim of the gastrular stage is to form two layers embryo. One layer will give rise to endoderm and mesodermal organ, and the other one will give rise to the ectodermal organ. You can see the examples of each layer and what organ it will give in future from the endoderm and mesoderm and ectoderm. Going through the process of formation of gastrulation, there is like 5 different mechanisms that gastrulation can use to do the process. The first one is invagination, which is the infolding of a region of cells to the inward, as the figure shows. The second one is the involution, in which there is interning or inward movement of an expanding outer layer so that they spread over the internal surface of the remaining external cells. The next method is ingression. It's a migration of individual cells from surface layer into the interior of the embryo. The third one is the delamination. Basically, it is a splitting of one cellular sheet into two parallel sheets. And the last one is epipoly. It is the movement of epithelial sheets that is spread as a unit rather than individually. And that's differentiated from ingression. This slide is an assignment. You need to answer all these questions and submit it to me by email. You can use internet resources, maximum 1 page A4 for the answers of the five questions. We'll talk now about cell specification, a process that happens once a gastrulation is forming, and it's the generalization of a specialized cell type, and this one as well is called differentiation. This differentiation is the last stage of a series of events that make the cell particularly committed to give certain fate. And this table just for knowledge to know how the type of cell and its differentiated product and its specialized function. For the commitment of fate of cells, the process of commitment can be differentiated into two stages. The first stage is a specification, in which the cell is capable of differentiating autonomously by itself when placed into a petri dish or a test tube. That's to say, in a neutral environment. And this type of cell commitment is reversible. While the second stage is determination, when the cell is capable of differentiating still autonomously, even when placed in another region of the embryo, that's mean non-neutral environment, and in this case cell commitment is irreversible. There are 3 major strategies of commitment that can embryo do, either autonomous or conditional or syncytial specification. For autonomous specification, and this occurs mostly in invertebrate, the blastomeres, as we said before, inherit a set of transcription factors that exist in the egg cytoplasm. For the conditional specification that happen in vertebrate and the few invertebrate, the ability of cells to achieve their fates by interaction with neighboring cells. So that means the cell to reach its fate, it needs interaction from the neighboring cells, which is totally different from the autonomous. The blastomere's fate depends on the position in embryo and is specified by paracrine factors secreted by neighbor cells. So you can see here that if we have these blastula and you remove some cells from the normal back cells of the blastula and transplant it into another region in another blastula, you will see that the cells which originally will form the back of embryo will form the belly tissue. That means that the fate of the cell have been changed due to its location. In another example, you can see that they use a glass needle to remove the cell at all and making a hole in the blastula, but still there is a normal development and there is a compensation of the missing part. That means that cells uses the new position to compensate or condition the specification. Dirichlet was the first scientist. He's a German scientist who provided the first experimentally observable evidence of conditional. He said that interactions between cells determine their fate rather some cytoplasmic factor. Still, the cells have cytoplasmic factor, which is the transcription factors or morphogenic determinants, but the interaction and the paracrine secretion between the neighbor cells enables this conditional specification. In this example, he took the 4-cell stage of an invertebrate animal. He removed the fertilization envelope and separated the 4 Blastomere stage into separate 4 cells, and left these 4 cells to continue development. He found that 4 same larvae have been produced. The only difference that the size is different, and they look the same as the original one which developed from the 4 Blastomere stage. This means that every cell behaved as a single cell, as a single embryo. Okay, the only difference between the normal Blutus larva and the 4 other Blutus larvae is the size. Again, Dirichlet continued to explain that he took the 8 Blastomere stage and removed the first top 4 cells, and the other separates the upper animal pole cells and lower vegetable pole cells. He found that the lower one developed into Blutus larva that normally half the size, while the upper half developed the part of the animal which will give to the animal the cilia that help this larva to move. In another example of the same experiment, he separated in another half in which each half will have 2 cells of the animal pole and 2 cells of the vegetable pole. Both of them develop into Blutus larva. That means that the neighboring cells controlled the 2 halves to be the same, while the other cells there is since in the first part, the first figure, you have the animal pole, all the cells have the same cell type, so each one each half in the first figure give different embryos, while in the other one, since there is interaction between the animal pole cell and the vegetable pole cells, they give whole Blutus larva, but it's half the size of normal. So we'll talk about how the morphogen gradients and cell specification. Again, morphogen gradients, it is the biochemical molecules that can be transcription factors that is inherited from the mother ovum of mother egg cytoplasm. These are transcription factors produced within the cells or paracrine factors that affect neighbor cells. If the cells are uncommitment cells, this is exposed to high concentration of the morphogen, are specified as one cell type. When this morphogen concentration drops below a certain threshold, the cells are determined to another fate. When the concentration falls even lower, there is a third fate can be specified. So in this simple example, if we consider if we consider a cell color which is blue, white, and red, and we consider there is a gradient of the morphogenic factors starting from the source high concentration into the sink lower concentration, so you can see that when the source have a concentration, when it decreases until it reaches the threshold of blue, the color change into white, and when it reaches a threshold of white, the color change into red. That means the color change it's like a reflection of the concentration of the gradient. In another example, if you look to the flag, the France and the US flag, if you take switch parts of the flags of the France between the France and USA flag, you will find that the region that it's transferred from the American flag to France flag developed into the same position stars as it's on the original flag. That's mean it as if it took the memory of the region of the same flag, while the other one, when transferred back, it developed the same part of the original France flag. So these reciprocal transplants develop according to their final position in the donor flag. Okay. The third type of specification is happening is insects is called syncytial specification. And what's meaning of syncytial? Syncytial, that means you have like a bag that's full of cytoplasm and there is a lot of nuclei with no cell boundaries. So it's like nuclei is swimming into cytoplasm. So the transcription factors or the morphogen are all over the cytoplasm and surrounding all the nuclei. So if you can see in the larva of the insect in the first figure, you can see you can mainly have two morphogenic transcription factor, which is the bicoid and caudal. And the higher concentration of the bicoid is in the anterior, while the higher concentration of the caudal is in the posterior. And there is a gradient decrease as long as we move into the middle. So in this case, the concentration and the ratio of the two transcription factor will determine the fate and the specification of the cells. And you can see that in the anterior part it's more bicoid and in the posterior part is more bicaudal. So that's why the bicoid is red and the caudal is blue. So that's why in the second figure you'll see that the nuclei turn to be red because it's affected by the bicoid, while the other one turn to be blue because it's affected by the caudal. And you will have in the middle, there is a mix of the true transcription factor which will develop into the other organs. That's mean in bicoid responsible for the formation of the head region, and the caudal is responsible for the formation of tail. Once the concentration fail in the middle, because the gradient is the minimum in the middle, the other organ are formed, either thorax and abdomen.